Hi, everyone, and welcome. I'm happy to be your host for today's webinar. My name is Dana Jaber, and I'm a Senior Clinical Affairs Scientist and a Customer Success Manager at Bionano. Thank you for joining us for the fifth session of the Bionano In Motion webinar series. By now, many of you are familiar with the series. It features cutting-edge research that showcases how optical genome mapping, or OGM, is being applied in the fields of hematological malignancies, constitutional disorders, and beyond. Today, we are delighted to welcome Dr. Wahab Khan, who will present on the impact and integration of optical genome mapping in hematolymphoid malignancies, with the emphasis on CLL and lymphoma, where there is growing interest in the use of OGM. Dr. Khan is an assistant professor of pathology and laboratory medicine at Dartmouth College. He also serves as a clinical cytogeneticist and molecular geneticist in the Department of Pathology and Lab Medicine at Dartmouth Health. His work focuses on interpreting human genomes from the chromosomal to the DNA level, with an interest in chromosomal structural variation and large-scale genomic sequence data analysis. Before giving the mic to Dr. Khan, I'd like to remind everyone that Bionano is a research use only company, and our products are for research use only as well. Now, without further ado, Wahab, thank you so much for being with us. The floor is yours.
All right, so as Dr. Jaber mentioned, thanks again for joining today and for inviting me to this seminar series that has seen now its fifth iteration. I'm really happy to be here. And I'm from Dartmouth Health, and the work I'm presenting to you today, it's about 45 slides or so, is a culmination of work done by a lot of our folks in our team. And I'm kind of walk you through some of that today. I don't have any conflict of interests to disclose or any financial relationships. Some of this work was supported by a sub-grant, NIH sub-COBRI pilot grant for translational OGM work in lymphomas. So I do want to note that. And let me just see if I can. So the kind of the overview of the talk today would be our experience in OGM validation. So, our journey and how we got here, some of the quality metrics that we take into consideration as we were building up this test and analyzing its performance. And then I'll transition more into some of the lymphoma samples, as Dr. Jaber mentioned, that we've worked up, as well as some of the leukemia interesting cases that we can show in the end part of the talk through some case examples. So the journey for us to current state goes back about a couple of years. We acquired the Sapphire instrument in the Department of Pathology in our institution at Dartmouth Health. We started some test familiarization stages, and we really didn't kind of kick off our validation in till the first part of Q1 in 2024. We were accumulating samples and freezing down samples prior to that. Into the validation, we put in about 80 plus samples. We relied a lot on the framework paper that was published very nicely in 2024 in Journal of Hematology that had a lot of good guidance in there. And then we also, allowed for our other ancillary sections like flow cytometry and hematopathology sections to be really on board with us to kind of supply any samples that we may have low volumes on. And then we started to summarize that data in the latter part of 2024. And, I'll show you some of the performance characteristics of that in the next few slides. But prior to that, I just wanted to take a step back and kind of touch on this aspect of chromosome structural variation, why we care about it. And over the years, I think, as you're all aware, I don't have to repeat it for this audience, that karyotyping being the gold standard and has allowed us for the last 50 plus years to be able to detect this variation in the human genome. And I think over time, with the advent of arrays and sequencing, and now with genome mapping and other third, fourth generation sequencing, this type of variation is only increasing. Our ability to detect the different classes of variants, insertions, translocations, inversions, with the advancements in technologies is opening up this search space, and we're finding very new things, recurrent things that we thought might have been rare, but yet might be under ascertained. And so if you look at if you take a historical lens and look at the 1,000 Genomes Project up until now with the pan-cancer genome analysis and the pan-genome analysis, the telomere to telomere assemblies, the growth has been exponential. And I would argue that, and others have as well, that structural variation comprise a larger amount of the genomic landscape that can be impacted. And if there's a gene in the middle of this SV that's either related to a germline event or an acquired event that gets impacted, can downstream result in issues. And if you look at sort of the growth of structural variants from the early 2000s up until present day, we've seen that there have been, with back arrays, oligo arrays, short read sequencing, and then population levels, large NGS and long read sequencing, this variation has increased 1,000-fold in the last 25 years. If you look at the growth of structural variants across different databases, that has also increased as well, and those are being captured in various different formats, minimum database being one of them. The prototypical example of an AML, where we know that there are often in the guidelines now new variants that have been noted, as well as variants that are cryptic. And so the rationale is sort of building up to the point where we need better tools to be able to detect these for downstream applications. So in summary, I think in terms of rationalizing this, the impact of structural variants both in the cancer space and the developmental space has been well noted. And so we need better tools to be able to ascertain these. And so I won't get into the details of what OGM is and how it works, but just again, that's been provided in previous talks. But just to note that it's a way to digitize looking at karyotypic changes, and it's a non-sequence approach. It looks at large-scale genome rearrangements. It starts with linearized ultra-high molecular weight DNA, which is labeled and then scanned on a chip, and then that can be then imaged. So in terms of getting into the first part of the talk, where we looked at different validation considerations, familiarizing ourselves with the protocol and being able to perform the concordance, the accuracy studies, and some of the performance characteristics. So I'll go through those in the next few slides. And so with respect to accuracy, we established this traditional 2x2 matrix where we had the comparator method to karyotyping in fish and our candidate method being OGM, and we achieved a fairly high accuracy of around 97.7%. We looked at over 100 different SVs, CNVs of different classes. This was a technical validation, and it noted that there are in some cases where the breakpoints were close to this pericentromeric region, or there were low-level clones involved. Those were sort of the ones that were more in the false negative group. And so our overall accuracy and positive percent agreement was around 94%, and overall accuracy was 97.7%. The second thing we looked at was specificity, so events that were truly negative at the whole, at the chromosomal or locus-specific fish level. In some cases, we had some heme microarray data as well, and we wanted to make sure that those events that exhibited diploidy or no net gain or loss or rearranged segments were in fact captured as negative findings on mapping as well. And so that's what you're seeing here. Where we didn't really report out any false positive events, and 100% of the reportable findings by chromosome banding analysis in fish around a 95% confidence interval were concordant. For the third thing we looked at, as you might appreciate, our reproducibility studies. So we had just the one Sapphire, so we had to get creative in how we set these up. So we ran these in duplicates, either inter-run or intra-run, and as well as with different operators. And we were able to achieve, in terms of capturing the different QC parameters, and I'll talk about those in the subsequent slides, about within a 20% variance with respect to the map rate, the label density, the coverage. So in these cases, they were fairly, close to each other in terms of the coverage, the map rate, and the percent label variance and the molecule length. We also did move, in addition to the earlier parameters, we looked at limit of detection studies. We did mostly in silico mixing as well as in vitro mixing experiments. We started with baseline and then mixed a wild type into a well-characterized structurally rearranged genome, mapped genome that was run prior, unmixed, and created these mixes to then ask the question, can we still detect, say, for example, I'm showing an 821 translocation in AML. If we filter down those molecules, the proportions of the samples, can we still faithfully reconstruct that variant? And we were able to find that it was, down to about 0.080 VAF. We were able to show that it still exists. We also included deletions and duplications as another class of variant. And that's what you're seeing in this slide, where we were able to show in an undiluted form with 100% VAF, 100% undiluted down to a 95:5 mix, that as you go to additional low-level mixes of these dilutions, you can still retain that VAF, but then you start to kind of lose it around, sub 5%. And these samples were selected with the same sort of relatively MQR, molecular quality reports, so that you're not mixing things that are, totally off in terms of their initial QC metrics. So that's an important consideration as you start to build your LOD data. So as we started to establish that, we wanted to then work out a workflow. So based on N of 1 instrument, if we were to, right now, if we extract, for example, on a Friday, we let it stay homogenize over the weekend and label Monday, the early part of the work week, and then we chip load on Tuesday. We have another iteration of this now that we're running an additional set of samples where we start the extraction on a Monday, more or less, and then let it homogenize, label on Thursday, and it gets scanned and load over the weekend. So in this way, we're able to run about a dozen samples a week. We're able to also do this sort of Monday, Friday setup with having a backup for each case. If we're able to, that would be ideal, and that's what we aim for. So we stabilize our samples and keep them sort of frozen as a backup in case needed. So now I'll switch over to key sample run metric considerations with genome mapping, and just to kind of point out, and you may already know this for this crowd, that we're looking at molecule length. We're trying to capture the on-target rate, the coverage, as well as the label density. And prior to that, during the initial wet lab procedures, the critical, some QC stop points for us at least are, how much of the white blood cell count that we're getting, the total white blood cell count, the concentrations being recovered post extraction and then post cleanup and labeling, and then we remeasure again as we go forward. And so I won't spend a lot of belabor the point here again, but just by definition, for those who may not know, the N50 is the molecule length. The label density is the 6-mer motif that occurs across the length of the chromosome. Every 100 kb, we expect to see about 14 to 17 of these labels. The average map rate is the on-target sort of percentage of molecules that align back to the reference, and then the effective coverage that is achieved following the complete mapped molecules. So in our hands, based on the 82 samples that we put into the initial validation, we were able to show the following sort of cutoffs, establish the following cutoffs, and we're still evaluating these as we build more samples into the research cohort or the clinical research cohort. So our map rate has been fairly stable around 80 to 90%. Our coverage has been north of 300x. Label density has been good, and the molecule length's been good. We do QC flag these in instances where it's below, and each lab has to kind of come up with their own parameters. There are some recommendations in the framework paper, but when it's sort of 2 of these are out of bounds, we have to look at the data and review it to see if it's still analyzable. Kind of the deal breaker on our end, at least, is if the coverage is really, really low, below, say, 300x or 280x, we kind of scrutinize that sample a little bit more and look at the copy number variance, because you could potentially miss aneuploidies in that case, which you can kind of mitigate by going back to a backup sample and troubleshooting that. But for the most part, this has been pretty stable for us, and we've gotten good QC parameters back. The other thing that we've implemented that has helping us, and we're trying to move this to a more automated dashboard type thing, is where we're starting to collect, when was the sample, things like you can capture the sample type, was it blood, is it EDTA blood, is it heparin blood? Importantly, when it was collected, and importantly, when it was stabilized. And so in our hands, we've in the validation, we demonstrated that anything up to 3 days, we could still kind of take and process. Beyond that, there's a risk for, attrition of some of the molecule length. But, this information is good to capture, so you can kind of go back to it and reference if needed. And then there's some other parameters here that are also captured during the wet lab stages of the process. And then these are the QC parameters that I talked about that are important to kind of track and capture to see if there's any, or rule out if there's any drift in the sample QC over time. So this is an example of a good quality run, and this is the usual scenario for us, where we're getting good map rate, good coverage, and we try to collect around 1,500 to 1,600 gigs of data for our heme conditions and as well as the lymphoma workup. In this case, if you have, and I'll show you a couple examples where it may be a little bit on the lower end. So this is low labeling density. If you remember, it's about 14 to 17 labels we want to see on average per 100 KB. And in this instance, we're seeing a little bit lower than that in flow cell 2. And so this can potentially impact your map rate as well. So this is something that you'd want to reprocess potentially from the second day 2 steps. Visually, it could look like this, where there's this unequal labeling pattern on the chip. And I'll show you an example where it's more uniform. So, there could be things where there might be an inhibitor in the sample. We did increase our cleanup steps, protein K digestion steps for DNA extraction and labeling on both days. Sometimes if the concentration is out of bounds, this is what's been recommended in the framework paper. We've been able to actually go a little bit higher than that and still demonstrate good QC, and then a little bit lower than that. But if it's out of bounds a lot with respect to this range, it may be that there's inefficiency in that enzyme labeling that DNA efficiently. And just some reminders that no-brainer reminders where storing the reagents in the proper temperature, and not making sure the business end of that kit, the enzyme, is not gone, rendered inactive. And you could have more sort of hardware related issues that require more of a preventative maintenance call, and that could result in also sort of seeing this low labeling density if that's seen over time. This is an example of where you might see a sheared DNA phenotype. So because we're extracting ultra high molecular weight DNA, you have to be very, very careful that mechanical shearing is minimized, and the usage of wide bore tips are implemented, as you might be aware. And so if you see that occurring, again here in flow cell 2, for example, we're seeing sort of this N50, the median length, being a little bit lower, or lower than what we want to get, around 230 or above. And so so this is a good scan pattern where it's uniform and looks fuller, and the molecules span the length of this sort of window. It's not drawn to scale, and this is somewhat of a more fragmented nature pseudo drawing of what that DNA might look like. And so just as a reminder, that stabilizing that sample in time, and this is where tracking your sample collect to sample stabilizing matters, and just if you're adding buffers, you angling the tip away from the DNA, improper, no vortexing or fast pipetting. We do keep a backup aliquot, and we can go back and re-extract. So in this instance, we would probably have to go back to the original frozen aliquot and redo the extraction. So just wanted to kind of note that here. The wide bore tip is important to use, and you can kind of get these from like local vendors in your areas. Okay, so having gone through some of the validation work and some of the QC parameters that we are monitoring for this assay, I wanted to kind of switch gears and talk a little bit about some interesting cases that we've worked up in both the lymphoma and leukemia space, and kind of walk you through some of the case examples. So this was part of a small grant that we got at Dartmouth Health that allowed us to look at some lymphoma samples, some excisional biopsies that were later on fresh frozen and then worked up. And just to kind of back up for a minute and just assess it from a point of perspective of a global burden. So lymphoma, in terms of cancer-related deaths cases, ranks one of the top 10 incidence-wise. In terms of its morbidity or mortality, it ranks close to the top 10, ranked 11th actually, and it's a pan ancestral, or it affects sort of the other all types of ethnicities or ancestries across the different continental representations here. The prevalence is fairly high in the Asian subset, as well as the European. And as a reminder, just to be aware that this is a disease of the B cell or T cell process. It can lead to accumulation of neoplastic cells in the lymph nodes, and it comes in different stages of aggressiveness. So you could have localized disease. You could also have instances where it's more widespread. So it's now spreading to different organs or multiple lymph nodes. And so it's something that we currently monitor or work up with a karyotype, for example, or molecular workup. But being able to survey this, or an FFPE, foramen fixed sample, so we're wanting to look at the high grade and the low grade lymphomas, and those are important, have important characteristic markers in them that are important to track and identify early on. So in our research cohort, we included samples across both follicular lymphomas and diffuse large B cell lymphomas, high grade lymphomas. Some of these were surgical specimens, so they didn't know exactly what kind of the diagnosis would be. So it'd be kind of a rule out lymphoma specimens, as well as small lymphocytic lymphomas, and some CLLs were in there as well. We also included some T cell lymphomas in our cohort as well. The wet lab workflow here was a little bit different, and in that the sample, the tissue biopsy, once received in our gross lab, would be subjected to a flash freeze, stored at -80, and then we would collect it from there and then process it. In our hands, at least, we noted that about 5 to 10 milligrams of DNA, starting DNA, would be important or sufficient for extraction, and then we would then homogenize that tissue using a tissue ruptor, and then it would then follow through with a tissue DNA isolation protocol, which is slightly different than the sort of the bone marrow and the blood-based protocols, but then the downstream labeling is still the DLE enzyme-based labeling, and then scanning on the Sapphire. These are our initial quality parameters for the 25 of the lymphomas we've run in this research cohort, with showing you the label density, the coverage, and the map percentage, as well as the molecule length on the y-axis here and the samples on the x. We did lose 5 samples to a low coverage and map rate, but in these instances, we were able to demonstrate for the samples that went into this cohort fairly good, 300x to about 400x coverage. There were some outliers here. Fairly tight distribution of the labeling density. Map rate was north of 70%. This is the threshold line, and then the coverage was north of the molecule length was above 230 kb or 230 kb. Yep, for most of these samples noted. So in a subset of these cases, what we thought was very impressive to see was the ability to recover findings or results in cases where there weren't any dividing cells. Often it's the case with some of these lymphoma samples that you do tend to get, or tissue biopsies, that maybe there are not enough mitotic divisions. So in about 25% of those cases, 20 to 25% of those cases, we were able to show with OGM a pathognomonic sort of rearrangement. So then this is one such case where the lymphadenopathy was included in our cohort. We did not see any dividing cells on observation, but after doing genome mapping, we were able to show in this case a canonical immunoglobulin heavy chain BCL2 rearrangement, which you see here, and then as well as some aneuploidies across the way. And just to kind of orient the audience, that what you're looking at is a circles plot, and you may have seen these before, of course, where there's the ideograms, the genes in the regions where there are blocks or centromeres and telomeres where their label density is sparse, and the structural rearrangements in this ring that are below a certain KB cutoff, and then there's the larger copy number aneuploidy type events in this ring, and then the innermost ring are your translocations. So this magenta line is showing you that the 14q3234 region that's distal here is juxtaposed to this chromosome 18 region, and in all these instances, the chromosomes are sort of laying on their left to right, P arm to Q arm, and the red here represents the centromeres. So yes, so IgH BCL2 rearranged along with various trisomies, trisomy 12, X, and 7. The blue is synonymous with the gain. The red is synonymous with the loss. And then we can click on this, and when we did, you can kind of see the map. The IgH locus, as you might be aware, or any of the immunoglobulin heavy light chain, kappa chain loci are fairly large. The breakpoints are fairly challenging. They occur in non-coding sequence, so the DNA-based assessment of these rearrangements is really important to kind of capture those breakpoints in those regions. And so what you're seeing here is the reference molecule on the top, chromosome 18, the reference molecule on the bottom, 14, the test molecules in the middle, and it's mapping to one part of the derivative 18 and the other part of the derivative 14 here, with the nice ability to be able to look at the genes impacted simultaneously, which are often covert on a G-band analysis. So you have to kind of do that reflexive testing. And then further down here was the IgH locus. These are the IgH loci that are further down below. So we can examine this call. We know that there's enough labels on each end of the break. It's a high confidence call. There are a lot of over 100 molecule supporting reads. We also noted, so as it goes, we can always find additional things with optical genome mapping, and in those cases where there's a hematolymphoid gene involved, we were able to ascertain that. In this case, there was a MIB deletion, which is an oncogene associated with leukemogenesis, and so that's being shown down here in both the copy number track as well as the structural variant track. And it shows you that there's an abundance of labels here that are missing. So it was on the order of about a megabase or more, I believe 4 megabase loss in MIB. And then again, you can see the allele fraction down here, which is we saw it at about 0.490, and at a high confidence call. So here's another example of a B-cell lymphoma that was really worked up as high grade. Again, we weren't able to achieve dividing cells in culture, but we did have some fish data for this, where using a break apart probe, we saw additional signals in loci that were targeting BCL2 and BCL6 on chromosome 18 and 3 respectively, and that tracked with the gains seen on of the entire chromosome seen on OGM. So this doesn't really give you that full copy number or the aneuploidy event because it's localized, targeted, whereas you can kind of get that through this. And so this was a case where there weren't as many SVs driving this event. There was this one call on X and 4, but didn't really have an important hematolymphoid gene involved at the breakpoint as far as we could tell. So maybe a kind of a passenger event or some maybe uncertain finding. But we did note that in this case, these aneuploidies were noted in this lymphoma sample. So, and this has been described in the literature where there's, if there's an absence of recurrent translocations of note in certain B-cell lymphoma processes, there have been described partial or full aneuploidies that we should be aware of. It's not always easy to add all these different types of markers on a FISH panel. So I think it was nice for us to kind of consolidate that and show that in one go on the circles plot. And some of these have been implicated in terms of the gains that we saw in as early findings in B-cell lymphomas. Okay, so I'm going to shift gears a little bit and show you a more complex case that was also worked up as a T-cell process, a T-cell leukemia lymphoma. This was a bone marrow specimen concerned for mature T-cell lymphoproliferative disorders. And the karyotype came back and showed a lot of uncertainty, a lot of add chromosomes, as you can note here, a lot of composite karyotypes in this. So outside of it just being kind of rearranged or highly rearranged, we couldn't really say much with respect to what's happening here. We did take this fresh frozen tissue out, and this shows you the G banding output on the chromosome bands, which kind of shows you almost every chromosome has a rearranged morphology. And so in keeping with that, we were able to successfully map this, and it demonstrated this complexity here on the left panel here of the circles plot. You can see we were then able to, from this, filter down using lymphoma-specific region files and genes to kind of get at some of the more interesting events. And what we found, interestingly, that there was an ATM loss, there was a gain of 8Q, loss of 8P, which was in keeping potentially with this ISO 8Q. If you go back to the karyotype, you can kind of get a sense of this. This band pattern here looks a little bit odd, but you can in retrospectively kind of go back and kind of match it up. ATM there was somewhat of a focal loss on ATM that was below the limit of G bands, and that's an important marker in this TPLL prolymphocytic leukemia. We also were very interested in knowing if there was a rearrangement in the TCL1A locus that occurs at chromosome 14q32, and that did not have a recurrent translocation that's often found in this, but it is one of the major criteria, but we were meeting some of the minor criteria for this particular case. And then we again saw these partial and full chromosomal aneuploidies across the genome. Of note, again, was sort of this 17p loss with almost this chromoagenesis or asynthesis-like pattern seen on 17q, as well as, here's your sort of focal loss of ATM, and then this was just added into with the TP53 being lost. So just to collect the list of aberrancies in this particular T-cell lymphocytic leukemia, ATM loss, TCL1 being rearrangement negative with the 8Q gain, the complex karyotype, and on sequencing, we did see a JAK1 missense variant. So it all sort of fit this criteria of it being this TPLL, and then, kind of going from, this to being able to kind of compartmentalize some of these findings into this was really encouraging to see here. And so it was favoring a TCL1 negative TPLL. So I'll move now a little bit into our CLLs outside of the lymphoma work. We've been doing this for the longest. So we've worked up over now 150 CLLs by OGM that we've been running. And I'll show you kind of the subset of those 50 or so samples that we've processed and some of the additional relevant pathogenic findings. So in totality, of course, we're always, as you run these region files, you can kind of start to pick up some of the oncogenes and tumor suppressor genes. Some of the ones that were recurrent in the cohort that we looked at are noted here. Of course, the 13q loss with this long non-coding RNA was prevalent, as is well known in CLL biology. ATM loss, RB1 loss was also seen in some of the larger type 1 deletions. Oncogenes that were impacted are noted here. CCND1 rearrangements are tricky. Sometimes they don't necessarily create a direct fusion event. There could be, in the breakpoints again are sort of like immunoglobulin chain rearrangements where they're in this larger regulatory region. So those were really nice to be able to start to capture. And then some of the more rare, perhaps recurrent translocations that we saw. And I'll show you a couple of examples of those. So this was a case where we've picked up a rather rare, possibly recurrent translocation. Sometimes it's hard to know if they're in fact rare or if we've just been under ascertaining these over time. This had a monotypic B cell population detected. The differential had this atypical CLL, and we noted here a sort of a double hit. It was really interesting to see this. Though it was sort of this translocation 214 and then translocation 1419. With the the BCL11A gene was involved with respect to immunoglobulin heavy chain gene, and with respect to 19, BCL3 was involved with juxtaposed to the IgH locus. And the karyotype corroborated that, although morphologically or cytologically corroborated that, where you've seen the derivative 2 here involved with part of one of the 14s, and then you have the derivative 14 here, the other homolog involved with the 19. So we're able to phase this information on the metaphase, of course, but you can kind of infer some of that through the VAF that were slightly different here, but it's an inference at best. And so being able to detect these 214, 1419 translocations, we don't we don't really look for these in CLL or think about these too often on a FISH panel at least, but BCL11 IgH has been observed as an initiating event in CLL cases that harbor this translocation. So it was nice to be able to kind of go from here to kind of arrive at some of the gene level information. This was another case in CLL where we were able to pick up another rare event where CCND1 IgH rearrangement was negative. That was being suspected on a different and other probes on this panel for the FISH were negative. But we found an interesting rearrangement with the chromosome 22 juxtaposed to chromosome 11, and the gene here was IGL. So it's 22q11.2 immunoglobulin light chain cluster of genes, and it invariably sort of became, instead of a CLL, this turned into a mantle cell lymphoma with respect to this particular driver event involving CCND1 and IGL. So has been very well recognized in mantle cell biology, B-cell neoplasia, and has been published before. And this kind of shows you the karyotypic view of that, with the 22 showing the attachment of the additional G dark, G light region coming from the 11, and the reciprocal translocation here with the 22q. And so we went on to kind of put this out and publish it, and it was an interesting, was a nice finding that we don't often survey these regions that are non-heavy chain rearrangements, but it was we were able to show this in this one case. And what we're trying to do with this, as we accumulate more of these sort of rare rearrangements, we're trying to plot it as time to results and with respect to prognostic to be able to see if these are relevant in that particular entity. So the y-axis here shows you the time to results in months. The x-axis here shows you the samples in this cohort. We're seeing some of those that were picked up with OGM being tracked as important findings that had an earlier time to result versus some of the ones here that are being tracked late. So I think this is a work in progress, and we're trying to build on this information a little bit more. So I'll move a little bit into the myeloid disease, and we'll kind of finish there. Hopefully we're doing good on time. So this was an MDS to AML conversion where we saw, in this case, a MECOM inversion, but that MECOM inversion wasn't the typical one that you see with RPN1. That's sort of the paracentric inversion 3Q. This was a much narrower inversion, so it was missed on karyotyping. It was missed. It was not. It was suggestive on FISH, but not very obvious. It was not directly identified. So this was the narrower inversion. So I kind of did a partial digital karyotype, if you will, with a partial karyotype here of a traditional MECOM inversion, which is a bit wider in its map. And you can kind of see that this is this one being shown in the drill down map view, and you can see the MECOM gene up here, and it was juxtaposed to this novel or new partner gene on 3 as well, within the same cytoband, in fact, that is noted here at the bottom, IFT80. So it's disrupting MECOM on the dual color dual fusion probe that we run for this. We did see extra signals for MECOM, so that was highly suspect, and this was seen in a substantial number of cells that were analyzed out of the 200 that we typically do. So it kind of tracked with this being disrupted and broken and kind of causing an extra signal to be present. The karyotype was apparently normal, didn't see it. So had we not done this to kind of interestingly run this case, we wouldn't have be able to ascribe it to a sort of an AML rather than an MDS category. So those were sort of the cases I wanted to go through. I wanted to just in the last couple of minutes or so just switch gears and kind of say that we're in this phase 2 now where we've are applying OGM more readily to the CLLs and complementing it with some legacy tests. StatFish is still happening. Phase 2, we've started to do this for MDS and AMLs, and to be able to then comparably do the FISH to be able to get the stat in the low-level clones, baseline karyotyping for new AMLs. We want to get in, move further into the adult ALLs. Those are often SV heavy samples, so I think OGM would be a good approach there. And then the B cell processes, the lymphomas, and possibly the myelomas as well. Just to kind of give a, we were able to show a lot of this using a single assay and identify some of these rare events that were concordant to the karyotyping and the FISH that I showed in earlier slides with the validation. But, just to kind of note that complex ploidy is sometimes harder to pick up, breakpoints near centromere, subclonal events. VIA, which is the next generation software to visualize OGM data, has a better filtering characteristics and heuristics to be able to pick up some of that complex ploidy. So I think those are some of the cases that we want to recheck there. Okay, so with that, I'll just summarize by saying that in a lot of cases, when we're looking at structural variants that are very much important and emerging, as I noted in the earlier slides, we often have a multiple technology approach that one can take historically. But I think being able to kind of look at some of these with newer tools that we have now, such as OGM, can provide a whole genome map, and it does very well at detecting some of these structural events. Often you need to be able to be aware of the assay implementation considerations. So considering having a pre-analytical, analytical, post-analytical checklist, having kind of your performance characteristics being documented over time, especially time to collect, time to stabilize, so checking the pre-analytical sample considerations before you're running them through the workflows. And so while no technique is perfectly able to detect everything, we're trying to reconstruct the karyotype here in a digital way. There's been many published studies that have shown that the approach here is a powerful way to look at least hematological malignancies in cancer cytogenetics. There's been, and I don't have to tell this crowd, but the International Consortium has published a framework paper that has allowed for further aid in labs implementing this workflow. So I wanted to, as a final slide, just give a big shout out to all our lab members, from our undergraduate students to our fellows, our admin, our core cytogenetics and molecular folks, our faculty members and flow and histology, as well as our HemePath faculty. And so this is a bit dated slide. I don't have the updated version on here, so apologies to anyone who may not have made it to this, but we will update this. Just a big shout out to our hematopathology unit and our fellows and our previous fellows and future genomic scientists that have helped us. And without them, I wouldn't have any slides to show you today. So thanks so much. And as a plug for the user call, we recently, Dartmouth and Harvard, Brigham and Women's Hospital, Dr. Michael Phillips and Philip Michaels and I, led a U.S. OGM user group meeting. Finally, it happened in mid-September, and that was we discussed some hot topics and just wanted to kind of in OGM workup. Wanted to just kind of plug that there's a lot of traction being made through different groups throughout the community, the OGM community throughout the world, the Spanish, Canadian, French, and European groups. So please check these out as you get the chance.
Thank you again, Wahab, for your time and for your expertise in shedding the light on the use of OGM. And before we conclude, I just want to take a moment to thank all of our attendees for joining us today. We hope you find this session informative and valuable. Have a wonderful day, and we look forward to seeing you in our next session.
Thanks everyone for attending, and thank you for the opportunity to speak today. Thanks a lot.
Thanks, Wahab.